Hello dear students, welcome back to Biovidyalaya. I hope you all are doing very well. Today in this lecture video we are going to discuss about one cell biology technique, fluorescence recovery after photobleaching, FRAP. FRAP is fluorescent microscopy technique, and based on this technique you will get both part B and part C questions in your CSIR NET examination. So, at the end of this lecture video, we will discuss some CSIR NET previous year question based on FRAP. So, please try to watch this video till the end. So, without further delay, let's get into today's class. FRAP means fluorescence recovery after photobleaching, and this method is used to determine the movement of cellular molecule. That means it is used to study the mobility of biomolecule. Okay. Our plasma membrane is fluid in nature because lipids and proteins in these monolayers are free to move. And let's see how what type of movements are possible for these biomolecules. There are three types of movement: rotational movement, lateral movement, and transverse movement. Transverse movement is also known as flip-flop movement. Okay. Rotational movement means rotation around its own axis. That is called the rotational movement. This molecule can rotate on its own axis, like how Earth rotates on its own axis. That is called rotational movement. And lateral movement means one molecule can move to another place on the same monolayer. That is called lateral movement. See, this molecule can move to this position or this molecule can move to this position. That is called the lateral movement. So, one molecule can move to another place in the same monolayer is called lateral movement. Transverse movement means a molecule present in one monolayer can move to another monolayer. Same way, the molecule present in this monolayer can move to this monolayer also. That is called the flip-flop movement or transverse movement. That means movement between the leaflets. Okay. And lipids can show these three movements: rotational movement, lateral movement, and transverse movement. But proteins can only show rotational movement and lateral movement. They are not able to do transverse movement. Okay. So, this FRAP technology is used to study the lateral movement of lipids and proteins in the plasma membrane. Okay. So, FRAP is a widely used technique to monitor diffusion of lipids and proteins in biological membranes. It quantifies lateral movement of lipids and proteins within the plasma membrane. You have to remember that first FRAP was developed to study lipid molecules in the cell membrane, like how it is organized and how it is moving, like that. But now it has widespread application, like for studying proteins, how protein interaction is happening, how protein is binding, how it is interacting with cellular components, and also even it is useful for studying how molecules are moving inside the cytoplasm. Okay. So let's see the details of FRAP technology, like how it is working. This is cell and this is membrane. This is cell membrane and this is lipid or protein. Okay. In FRAP, first what we will do? We will label the membrane with fluorescent molecule. Okay. Or we will tag it with fluorescent molecule. For example, GFP. GFP means green fluorescent protein. Okay, this will give green fluorescence. Okay, so we will first tag protein with GFP or any other fluorescent molecule, and then we can observe it through a fluorescent microscope and we can see that a uniform fluorescence. Okay, and after that what we will do, we will select one area, and then we will focus laser beam to that selected area, and because of that laser beam this area will become non-fluorescent, because this laser beam will destroy fluorescent capacity of that molecule. Okay, non-fluorescent. So the second step is, this is second step and this is first step. Okay, so second step is bleaching specific area of the membrane with laser beam so that area will become non-fluorescent, and this process is known as photo bleaching. Photo bleaching with the help of laser beam. Okay, so this selected area will become non-fluorescent as compared to the rest of the area, and then we will wait for some time, and we will observe the cell through a fluorescent microscope after some time, and then we can observe that some bleached area recovered its fluorescent capacity. Why? How it is possible? This is due to the lateral diffusion of labeled molecule to the bleached region. Here you can see that there is non-fluorescent protein is there, and here a fluorescent protein is there. This is due to the lateral diffusion of labeled molecule to the bleached area, and that's why this bleached area again become fluorescent. Okay, and this recovery is known as fluorescence recovery. Okay, fluorescence recovery. Fluorescence recovery after photo bleaching. Okay, so first what we will do, first we will tag membrane with some fluorescent molecule or labeling the membrane with fluorescent molecule. Then we will bleach specific area of the membrane with the help of a laser beam so that that area will become non-fluorescent. And after some time, due to the lateral diffusion, the labeled molecule diffuse into the bleached area, and that bleached area again become fluorescent, and that is known as fluorescent recovery. Okay, the rate of recovery of fluorescence is associated with the rate of lateral diffusion. Okay, that means the speed of recovery of bleached area is directly proportional to the diffusion speed of fluorescent molecule. Okay, this is the same thing what we discussed. Here you can see that two layers. This is bilayer, and this bilayer is uniformly labeled with red color fluorescent molecule, and this is side view, and here you can see the top view. So this bilayer is uniformly labeled with the red color fluorescent molecule, and here we are selecting one specific area and we are photo bleaching that area with the help of laser beam, and this molecule lost its fluorescent capacity, and this is bleached area or non-fluorescent area. Okay, here you can see that this area is darker as compared to the rest of the area. So this is bleached area. Okay, after that we can observe the fluorescent intensity, and we can find that some fluorescent molecule come into this bleached area, and at the same time some non-fluorescent molecule come out of this bleached area. So it is restoring its fluorescent capacity. Okay, so here you can see that eventually it attained the uniform intensity as compared to the first one A. The fluorescent intensity is almost same, but it is little bit less in D. Okay. Okay. So it is eventually restored its fluorescence. Okay. By seeing the fluorescent intensity, we can calculate diffusion coefficient. We can calculate diffusion coefficient. We can quantify how many molecules diffuse into and out of this bleached region within a specific time. Okay. So by observing fluorescent intensity, we can calculate diffusion coefficient. As I told you that the speed of recovery of the bleached area is directly proportional to the diffusion speed of the fluorescent molecule. Okay. You can remember that higher the diffusion coefficient, then higher will be the diffusion ability. Okay. Or diffusion speed. Higher will be the diffusion ability, or we can say that diffusion speed. Okay. If higher is the diffusion speed, then recovery will be faster. Faster recovery. Okay. See this graph. Here you can see that in y-axis, here fluorescent intensity is given, and here it is in x-axis time is given. Okay. And here A means when we tagging the bilayer with fluorescent molecule, here the fluorescent intensity is maximum. Okay, the intensity is 1. We can see that fluorescent intensity is 1 here. Okay, and here you can see that after photo bleaching, that fluorescent intensity will become minimum. Here it is approximately 0 only. Okay, and after some time you can see that the intensity is gradually increasing. Fluorescent intensity is gradually increasing, and finally at D it is reaching almost complete intensity as approximately 1, but it is little bit less than 1. Okay, and so we can say that this is the time at which it recovered. It is fully recovered its fluorescent intensity. Okay, I will draw one graph for molecule A and B. Let's imagine molecule A is a lipid and molecule B is a transmembrane protein. Here you have to remember that if a molecule is more bulky, less will be its ability to move. That means diffusion speed is less. Okay. So I will draw the graph here. Fluorescent intensity in y-axis and time in x-axis. Here 100, 200, 300 and 400, and here 0.2, 0.4, 0.6, 0.8 and 1. This is maximum intensity. Okay. Here I am drawing after photo bleaching only. Okay. This is for molecule A. Okay. And this is for, I will change the color. This is for molecule B. Okay. So here you can see that fluorescent intensity is reached maximum by this molecule A within a limited time period as compared to molecule B. Okay. So diffusion coefficient of this molecule is more as compared to molecule B. Okay. That you have to remember. The diffusion coefficient of molecule A is more as compared to molecule B. That's why it reached the maximum fluorescent intensity very fast as compared to molecule B. So the diffusion speed of this molecule A is more as compared to molecule B. Okay. Let's see the questions based on this FRAP technology. First question, this is the question from CSIR NET June 2016. This is part B question. This is very simple. Lateral diffusion of proteins in the membrane can be followed and diffusion rate calculated by option A, atomic force microscopy, option B, scanning electron microscopy, option C, transmission electron microscopy, and option D, FRAP, fluorescence recovery after photo bleaching. So which method is used for determine lateral diffusion of proteins in the membrane? Yes, this is FRAP. Option D is the correct answer. Okay. This is the question from CSIR NET December 2016. Fluorescence recovery after photo bleaching. FRAP is a method to estimate the diffusion of molecule in a membrane. Fluorescently labeled molecules such as 1. A receptor tagged with a green fluorescent protein GFP. 2. A receptor labeled with GFP which interact with the cytoskeleton. 3. A labeled lipid. 4. A labeled protein that bind to the membrane surface. Is photobleached and the recovery profile A to D were obtained to estimate their diffusion coefficient. So this is the graph. The following data were obtained. Which one of the combination is correct? These are the four options. From this four option we have to find the correct combination. Okay. So we can categorize this question under a tough question, and this is also a tricky question. For this question, smart way of answering is needed. So here in this first sentence, they given just the definition of FRAP and fluorescently labeled molecules such as first one is a receptor tagged with a green fluorescent protein. So receptor means that has transmembrane domain. Okay. So it is bulky and so diffusion speed is less. This is one. Okay. Second is a receptor labeled with GFP which interact with the cytoskeleton. So this is also a receptor but it is interacting with the cytoskeleton. Okay. Cytoskeleton means actin or intermediate filament. So this is also not able to move fastly. Okay. Because it is interacting with the cytoskeleton and diffusion speed of this molecule is less as compared to the first one. Okay. Next, third, a labeled lipid. Okay. Lipids are very fastly move in the plasma membrane. Okay. Diffusion speed or the diffusion coefficient is more for lipid. Okay. And the fourth one, a labeled protein that bind to the membrane surface. So these proteins are also easy to move. They are membrane surface protein. Okay. So let's see the graph. Here in this graph, B, A, D, C is given. A to D is photobleach and recovery profile A to D were given. See here which one is showing best recovery? B. The graph B is showing best recovery. After that A. And the less recovery is C. The graph C is showing less recovery. Okay. So, which fluorescently labeled molecule can easily move? Yes, lipid can easily move. So, it will be, B will be 3. Okay. And sometimes you will be confused with uh, whether 3, that means labeled lipid, or protein that bind to the membrane surface is easy to move. That time you can say that this is 3 or 4. Okay. And if it is 3, then A will be 4. Okay. If A is 3, then B will be 4. That you have to remember. Okay. So it will be 4 or 3. Okay. If you have confusion, then you can write like this. And see the options then. So let's see where it is given B and A. Here A is given 1. 1 is not possible. It may be 4 or 3. Okay. So we can, here B is given 2. It is also not possible. We can eliminate this option. Okay. So we can eliminate this option. Okay. So B will be 3 or 4, and A will be 3 or 4. Now we are checking options for that. Here A is given 4. So it is also possible, and here B is given 3. It is also possible. Okay. Here A is not given and here B is given. It is also not possible. So we can eliminate option 1 and option 4. Okay. Now we have option 2 and 3. Okay. So see this graph C. Here we can say that this graph is for the molecule that is moving very slowly. Which one is moving very slowly? Yes, the receptor labeled with GFP, which is interacting with the cytoskeleton. So this is 2. This is C is 2. Okay, so let's see. See, here C is given 3. This is not possible. This is not possible. So we can eliminate this option. Because 3 is labeled lipid. That is not possible for this graph C. So we can eliminate the option 3 is also 3 also. So the answer is option 2. B3 and A4. So B is 3, A is 4, and C is 2 and D is 1. Okay. This is the last question. This is also CSIR NET previous year question. Using FRAP, fluorescence recovery after photobleaching technique, diffusion coefficient of three integral membrane protein M1, M2 and M3 in a kidney cell is calculated as 1 μm per second, 0.05 μm per second and 0.005 μm per second respectively. Considering fluid mosaic nature of biological membrane and relationship of structural organization of integral membrane protein with diffusion coefficient, which protein will have highest number of integral membrane domain? Options are option A, M2 and M3, option B, M2 only, option C, M3 only, and option D, M1 and M3. So here diffusion coefficient of three integral membrane proteins are given. M1, diffusion coefficient is 1 μm per second. For M2 it is 0.05 and for M3 it is 0.005. Okay. So here, considering fluid mosaic nature of biological membrane and relationship of structural organization of integral membrane protein with diffusion coefficient, which protein will have highest number of integral membrane domain? We have to find which protein has highest number of integral membrane domain. Okay, you have to remember that the protein with more integral membrane domain has less diffusion coefficient. More the integral membrane domain, then less will be the diffusion coefficient. Okay, so let's see which protein has less diffusion coefficient. So here you can see that 0.005 for M3. So it has less diffusion coefficient. So this molecule will be more bulky. That has highest number of integral membrane domain. So let's see the options. Option C, M3 only. So this is the answer. Okay. I hope this video was helpful for you. If it was helpful, then please share this video with your friends who are preparing for CSIR NET examination. And also, don't forget to subscribe this channel if you haven't done it. Keep learning. Thank you.